Welcome to this afternoon's ATS Science and Innovation Center webinar titled Beyond Alum and Capsular Polysaccharides. My name is Alexa Pragman. I'm from the Minneapolis Veterans Affairs Medical Center and the University of Minnesota, and I'll be moderating today's event. Our speakers today are Elizabeth Norton from Tulane University School of Medicine and Jay Kolls, also from Tulane. Before we get started, I wanted to make you aware of another upcoming webinar in about 2 weeks on lung regeneration. Elizabeth Redente and David Largues will be presenting, and we hope you can join us then as well if the topic is of interest. Also, the ATS is hosting a virtual event August 5th through 10th called ATS 2020 Virtual. This is because the in-person meeting was postponed from last May. So please be on the lookout for additional details at the ATS conference website. And you can use this QR code when registration opens to register yourself for the event. And later today, the archived Science and Innovation Center webinar videos will be available here, and you can please look there for this recording later on. And as well, at the end, we'll ask you for any feedback, and you can access that here as well. So before I introduce our speakers and we get started, I just want to give you a couple reminders. Right now, because of the large number of registrants that we have, the audience is muted. So if you have questions as the speakers present, please submit them using the chat feature within the WebEx app. And as I mentioned earlier, this webinar is being recorded and will be available online starting tomorrow. So with that, I'd like to introduce Dr. Elizabeth Norton. She is an assistant professor in the Department of Microbiology and Immunology at Tulane University, New Orleans. She's a mucosal immunologist with extensive experience evaluating heat labile enterotoxin-based adjuvants and mechanisms of vaccine efficacy. She's pioneered novel LTA1 adjuvant for intranasal use in animal models. And today she'll be discussing mucosal adjuvants with us.
Thank you, Alexa. So as I said, here's Tulane University where we work, and I'll talk to you about mucosal adjuvants. So there's a number of vaccine challenges, and this is one of the reasons why we use vaccine adjuvants. These challenges include emerging infectious diseases. I don't think I need to tell anybody about that with the ongoing pandemic, but also re-emerging pathogens like tuberculosis, malaria, multi-drug resistant Staph aureus. There's also changes to existing pathogens that make them more virulent, such as epitope reassortment or recombination with influenza. There's issues with hosts with poor immune function, such as elderly adults or individuals with comorbidities or individuals on immunosuppressive agents, and also issues with the spiraling cost of healthcare delivery and even bioterrorism. So vaccine adjuvants, this actually comes from a Latin word adjuvans, which means helpful. And these are rare and unique compounds that stimulate immunity to vaccine antigens without promoting general inflammation and reactogenicity. In addition to magnitude of responses, they may help achieve a specific type or bias of immune response. The famous quote about adjuvants is calling them immunology's dirty little secret, which was by Charles Janeway in 1989, just because they are so kind of ubiquitously used in order to promote immunity, and they're not always very well understood. So only a few adjuvants are licensed in the United States. These include Alum and PL, which is a derivative of LPS, and ASF4, none of which are known for promoting mucosal immunity. So novel vaccine strategies, as I said, this is a focus area of mine where Our goal is to target immune responses directly at the site of infection or disease. And the rationale for this is because most infections enter through the body at mucosal tissue sites. So targeting the pathogen as it's coming into the body would be important. It also, if you do immunization through mucosal routes, provides an option for needle-free. So nasal or oral are great examples of these. And even the possibility of self-administration, which may be useful in either mass vaccination campaigns or depending on your target population. Another helpful aspect of adjuvants is they can allow for dose sparing, which can be important if you have limited amounts of the antigen for your vaccine. That is, you use smaller amounts of your antigen to reach more people when they're combined with adjuvants. The adjuvants I'm going to talk to you about today are LT-based adjuvants. LT is heat-labeled toxin from E. coli. This is an AB5 enterotoxin. And as you can see, here is the A subunit, here's the B pentamer. The A1 domain, which is enzymatically active, I will call LTA1 for the rest of my talk. And this is one of the adjuvants we're going to talk more about. And the other one is a double mutant. It has a mutation at arginine 192 and leucine 211. And this is called a double mutant of LT because it's now R192G and L211A. So these studies with LT and adjuvants go back well beyond my time into early 1980s. There were clinical trials done in the 1990s. Mutations were made because of the type of toxicity that was observed in humans with the initial native toxins. And DMLT is currently the most clinically advanced adjuvant from this family and has gone into both animals and humans. And a spinoff of this is our studies with LTA1 for an intranasal vaccine. So DMLT, AB5, as I said, with the 2 mutations. So the routes that you can use for this are oral and sublingual, as well as injected, such as intradermal or intramuscular or subcutaneous. There's also an option to do transcutaneous, where you actually place DMLT with the antigen on the skin and let it absorb through the skin. For LTA1, we have been developing this to be an intranasal or intrapulmonary adjuvant, although it can be injected. And this is because adjuvants that have this B subunit are not acceptable for delivery intranasally because there's been some link to neurological issues with the B subunit binding and inflammation from the vaccine. So the main features of these adjuvants, it's very simple admixing of the antigen with the adjuvant. There's no difficult formulation work that has to occur. You have enhanced antigen uptake at mucosal and parenteral sites. You get a multifaceted response, excuse me, TH1, TH17, and TH2, as well as IgG and IgA. And these adjuvants are really known for having this TH17 and IgA response. Excuse me. In addition, you can get promotion of mucosal immunity by either injected or mucosal delivery. So there's been many studies looking at DMLT and LTA1 in animal models. So these are a snapshot of these. These include for bacterial vaccines and for viral vaccines. I'm going to highlight some studies from ETEC with DMLT and tuberculosis today, as well as influenza for LTA1. And Jay is going to talk about his with Klebsiella pneumonia after my talk. There's no clinical trials with LTA1, but there have been several clinical trials with DMLT. These have found, as I'm showing right here, that by oral, sublingual, intradermal, or a variety of these routes, that DMLT itself is very safe in adults. It has also been used in infants in Bangladesh with no severe adverse events. It's been combined with a live attenuated ETEC vaccine. ETEC is enterotoxigenic E. coli, or a whole-killed ETEC vaccine called ETVAX. It's being used also in a subunit ETEC vaccine and in combination with the inactivated polio vaccine, as well as an HIV subunit vaccine. As I mentioned before, the history of these molecules really goes into safety and making sure the the molecule has been detoxified from the original native enterotoxin. And this is shown to be very effective with the two mutations, because what you see with the parent molecule LT, you get a dose-dependent increase in fluid secretion orally. When you have a single mutation R192G, you diminish that secretion, but you don't get rid of it. In contrast with the double mutant, you completely ablate secretion. These studies were in animal models, but this has also been found with humans who received the just DMLT in oral doses, and this study was published in 2013. And this, we've really linked to the stability of the A subunit of DMLT. It's slightly less stable than MLT and LT, and this results in reduced signaling in epithelial cells that mediates the secretion in the lumen of the GI tract. So as I've talked about, it's safe, but is it effective still? So the best study with DMLT thus far has really been this challenge study conducted in adult volunteers. These individuals received DMLT orally combined with a live attenuated ETEC vaccine called ACE527, which is a combination of bacteria that have enhanced expression of adhesion colonization factors. And when individuals received the vaccine, did not receive the vaccine, they had a challenge rate around 65 or so % to get diarrheal disease with this H1047 ETEC, wild type derived ETEC strain. If individuals had been vaccinated with ACE527, there was moderate protection. These are these white bars for % of subjects receiving severe or any diarrheal disease. And this protective efficacy was estimated to be 20.5%. And then when DMLT was added to the vaccine, the protective efficacy went up to 65.9%. So you can see just the addition of the adjuvant really enhanced the protective efficacy of the vaccine and made it much more functional for its design, its targeted design to protect against diarrheal disease. This is a newer study that we've done with DMLT for tuberculosis vaccination. This is where DMLT was combined with a fusion antigen and given to non-human primates intramuscularly with 3 immunizations. And then these animals were challenged a few months later with a lethal dose of Mycobacterium tuberculosis. And in naive animals, or animals who were vaccinated, or animals who were vaccinated with the antigen and with DMLT, we see enhanced protection of bacterial burden in the liver and also the bronchial lymph node, as well as in animals that received the antigen with DMLT vaccine, and we looked for lethality and survival. We see enhanced survival with if you receive the 5 fusion with DMLT intramuscular compared to naive animals, and an indication that this was linked to CD4 responses, particularly CD4 T cells secreting IL-17, which was much higher in vaccinated animals than naive animals. For LTA1, this adjuvant, the safety assessment is not really about secretion, but more about intranasal and neurotoxicity safety. So to show this, we did a study looking at whether mice, it's a behavioral study to see if the ability of mice to smell new odors changes after the mice have received a high dose of LTA1. And the reason why we did this study is because previous work had shown when you add these AB5 enterotoxins like cholera toxin, which is abbreviated CT here, that mice lose the ability to smell new odors like cheese and shrimp, things that they might be interested in. And so they don't come and investigate these odors. But what we find here is that, yes, the mice who are untreated come investigate new odors like cheese and shrimp. Mice who have received cholera toxin do not do this nearly to the extent as untreated mice, but mice who received LTA1 come investigate those new odors just the same way. So it's not neurotoxic, which is what our goal was in redesigning this adjuvant for an intranasal use. We also don't see any changes in histopathology in mice who received LTA1. There's no inflammation at the nose, the olfactory epithelium. We don't see any changes in particular cell types. So it's in all of our studies thus far, it's been extremely safe. And then the next question is, since it's safe, is it still working as an adjuvant? So for these studies, what we did is we took LTA1 in mice and we combined them with the human flu vaccine, Fluzone, which is abbreviated Fluzone, FC, with and without LTA1, and we immunized mice either once or twice, and then challenged them with a lethal dose of H1N1 influenza strain. And you see the addition of the adjuvant significantly improved survival compared to the Fluzone antigen alone. There's also a major reduction in just levels of viral RNA for the influenza virus. So here's our adjuvanted group compared to the Fluzone or naive animals. And this correlated with inflammation in the lung being reduced, looking at IL-6 levels, which would also relate to damage and just general inflammation from the flu infection. And this protection seemed to be mediated by both T cells and B cells because we saw increased levels of IgA and IgG in the lungs of mice, and this was dose dependent based on how much LTA1 was added to the vaccination. We also saw increased levels of interferon gamma, which would be a typical cellular response, and this we could see even more clearly if we looked at CD4 T cells in the lung expressed being activated. We saw a lot more of CD69, CD4 T cells in animals who had been vaccinated with LTA1 compared to the animals who just got the antigen alone. So we did one final experiment, which was really just to show the importance of either CD4 T cells or B cells in the LTA1 mediated protection from flu. So this is a little bit complicated, so let me walk you through it. We have either wild type mice who got injected with a control antibody, or mice who were vaccinated, which is all of these guys. This VAX is vaccinated. We looked for if we vaccinated mice, were they fully protected from weight loss and survival, and they were. If we then depleted CD4 T cells by injecting an antibody during vaccination, which will selectively deplete CD4 T cells for about 4 or 5 days, we saw, once again, complete protection. If we got rid of B cells by using a genetically engineered strain called MUMT, where the mice do not have the ability to have their B cells become mature as would be required for a vaccine, we see that there is almost full protection. These mice are almost fully protected from lethality, but they do have a little bit of weight loss. But if we combine the CD4 depletion with our genetic B cell ablation, we really lose almost all of our protection. So the LTA mediated protection involved both of our CD4 T cell and B cell components. So in conclusion, mucosal immunity is an important target for vaccination. DMLT and LTA1 adjuvants derived from heat label toxin of E. coli are potent mucosal adjuvants. DMLT is clinically advanced with safety and protective efficacies in humans. LTA1 is a newer adjuvant, but with promising results in mouse models for safety and as well as protective efficacy for flu. And as the data that I showed you, which is our newer data, just shows that this protection appears to involve a combination of both B cells and CD4 T cells. So thank you to everyone who made these studies possible. This includes individuals in my laboratory, as well as other collaborators at Tulane University, the Tulane University Primate Center, ARIS for the antigen in our tuberculosis studies, and our funding sources from the NIH. And with that, I'll conclude my presentation and try to stop sharing my screen.
Thank you, Dr. Norton. Our next speaker is going to be Dr. Jay Kolls. I'll introduce him now, and then we'll have time at the end for questions for both speakers. Dr. Jay Kolls is a professor of pediatrics and medicine and director of the Center for Translational Research in Infection and Inflammation at Tulane University School of Medicine in New Orleans. Dr. Kolls' work identified the critical roles of IL-17 and IL-22 in lung infection and demonstrated that antigen-specific Th17 cells can provide serotype-independent immunity to Enterobacteriaceae. His work has also demonstrated the importance of IL-22 in reducing lung injury in response to viral infection. He has pioneered the use of novel Fc fusion proteins for treatment of lung infections, studied CD4 T cells and B cell requirements for control of pneumocystis colonization, and pioneered the use of novel proteomic and transcriptomic strategies for antigen discovery that can be used for vaccines, therapeutics, or diagnostics. He's published over 350 manuscripts since 1992. Today he will speak on eliciting lung TRM cells with LTA1.
Great, thanks, Alexa. So let me try to advance here. Here we go. All right, so in terms of the disclosures, I have some consulting relationships outside this work. Obviously, none of this would be possible without grants from the NIH. And then the university here at Tulane has filed a provisional patent on this Enterobacteriaceae vaccine that I'm going to talk about today. So as we know, in the lung, there's three major effector cells, Th2, Th1, and Th17 cells. And what I don't have on here is Tregs or Th9 cells. Not to say that they're not important, but I'm going to focus on these three subsets. And the working model is that there's a division of labor across these cells, whereas Th2 cells are really critical for B cell help, as well as host defense against helminth infection. And then Th1 cells are really critical for intracellular pathogens. And then Th17 cells are thought to be critical for extracellular pathogens, particularly bacteria and fungi. So, I trained during the HIV epidemic, and, that taught us that CD4 cells were pretty important for pulmonary infections, because pulmonary infections, both with pneumocystis and streptococcus pneumoniae, were very common in HIV-infected individuals. Yet, when we think about vaccination, most of it's around targeting B cell responses and polysaccharides. So we've had a long-standing interest of, can we elicit lung CD4 cells as a novel way of immunization? And when I moved to Tulane 2 years ago, started a collaboration with Dr. Norton on LTA1 because of her prior work showing that it could elicit Th17 cells. And this is a cartoon that was published in Nature Medicine in 2008, and it showed this idea of this T cell talking to the epithelium here, and living in the lamina propria below the epithelium. And I showed this cartoon to David Woodland, who was a brilliant lung immunologist who was at the Trudeau at the time, and he said, well, why do you need a T cell to do this? Why couldn't this just be a fibroblast making this response? What's so important about a T cell? And that got us thinking. And so we took a bioinformatic approach, and we looked at the evolution of IL-17. And IL-17 is actually one of the oldest cytokines. The D isoform is the oldest. It's in Siona intestinalis in oysters. And in fact, if you infect oysters with Vibrio, a pathogen for oysters, the IL-17D transcript goes up in the oyster hemocyte. So it seems like a very evolutionary conserved response to infection. But what's interesting is humans have two isoforms called IL-17A and F, which are the predominant isoforms in T cells. They sit 50,000 base pairs apart from each other on the same chromosome, and so they kind of co-evolved with RAG genes. So you first see IL-17A in F in zebrafish, although there may be, there is an IL-17A isoform also in these lamprey as well. Now, important here is lamprey do not have RAG genes, so they can't recombine their T cell receptor. But zebrafish, all the way up to humans, we have RAG genes that allow us to recombine our TCR and make an antigen-specific response. The other thing that TCR recombination allows us to do is make memory cells that can respond quickly to a pathogen that we've seen, a related pathogen that we've seen before. So this informatic approach suggested that there had to be an evolutionary advantage of having IL-17A and F co-evolve with RAG genes, and we thought that evolutionary advantage was memory. And the way we tested this hypothesis is that we just immunized mice with heat-killed Klebsiella at the time, and we used this Capsular serotype 2 strain of Klebsiella. And what we saw was that most of the antibody response was directed against the capsular polysaccharide. However, the CD4 cell response was largely targeted to protein antigens that were conserved in other serotypes. So K1 as a hypervirulent strain of Klebsiella, K16, and that therefore these cells provided heterologous immunity against these other related but distinct capsular serotypes of Klebsiella. So obviously we're not going to be immunizing people with heat-killed Klebsiella, so we wanted to know, could we reduce this down to a subunit-based vaccine? And we knew that actually in this paper, we thought outer-membrane antigens were important. So if you take outer-membrane antigen preparations from bacteria, a lot of these responding T cells will proliferate in response to that. We also know that if you take outer-membrane vesicles, which package a lot of outer-membrane protein antigens, those also are very excellent vaccines. So we did proteomics on those, and one of the antigens we identified was this outer-membrane protein X here. And so that we prioritized that. And before I go into this vaccine strategy, I do want to highlight another paper that was recently published by Richard Flavel's group, who also uses heat-killed Klebsiella model to generate these lung TRM cells. And one of the interesting things is he showed that they require IL-7 for survival, and that IL-7 appears to be coming from parabronchial lymphatics, which is really quite interesting. So we immunize, in this model, we immunize twice, 3 weeks apart, and then we're immunizing with OMPX that's from a Capsular serotype 2 strain, and then we're challenging with a hypervirulent K1 mucoid strain of Klebsiella called KP396. And this immunization, this bacteria is quite virulent. So in unimmunized mice, even though we're inoculating with 10 to the 4th, we can recover 10 to the 9th, sometimes even 10 to the 10th bacteria in the lung after 24 hours after challenge. But in immunized mice, we get about a 6 log protection, both in the lung and the spleen. And what's interesting is that this vaccine induces parabronchial lymphoid structures, what we call IBALT. So you get local B cell and T cell areas, and we have evidence of class switch recombination of B cells with IgG positive and IgA positive B cells in these areas. So we wanted to compare these cells to the cells that are derived from heat-killed Klebsiella, since our lab and the Flavel lab kind of showed that they could generate similar responses. And then, so we did single-cell RNA-seq, and what you can see, this is a t-SNE plot, and you can see that the heat-killed Klebsiella cells, CD4 cells, significantly cluster with the OMPX LTA1. So, you can imagine this is a very complex, you have TR2, TR9 agonists, you have lots of different proteins, but the cellular response just with a subunit in the LTA1 adjuvant is very similar. And what's interesting here is this population here, if you can see my arrow, that you don't see here in the heat-killed Klebsiella, this is actually a TH1 population, but the IL-17 population is very similar. And in contrast, this is naive splenic CD4 cells, so they cluster completely independently. And then this is just a cytokine production by these cells. Here's the IL-17A cluster. There's this TH1 cluster here, and then there's a few cells that produce both IL-17A and interferon gamma in the CD4 population. Interesting, as opposed to in vitro Th17 cells, so it's well known that in vitro Th17 cells are plastic. So if you treat them with IL-12, they'll turn on interferon gamma. And these in vivo cells are not plastic. If you treat them with IL-12, they don't turn on interferon gamma, and they also in vitro cells will you can easily detect phosphorylated STAT4 in these cells after IL-12, where these cells you do not. And we think this is probably due to reduced levels of IL-12 receptor beta 2 on these cells. So they're quite different from in vitro Th17 cells. And they're also by V beta repertoire analysis, they're oligoclonal, so that they express high levels of V beta 14 in the immunized lung. Interestingly, we looked at TRI agonists to see whether they could also generate a similar response, but we failed to really, with both a TR2 agonist as well as a TR2/9 agonist, we don't see these same IL-17 responses that we see with LTA1, and these fail to really yield any kind of vaccine immunity in the lung. So we wanted to know are Th17 cells required, and one approach we took was to just knock out STAT3 and CD4 cells, and you can see that this dramatically reduces the number of IL-17A positive cells. We also get less T cells in the lung when we knock out STAT3, because STAT3 is a survival factor for Th17 cells, and this significantly abrogates the vaccine protection. So there's higher CFU when we knock out STAT3 and CD4 cells, as opposed to the littermate controls, Cre-negative littermate controls. So it appears that STAT3 is essential for this vaccination. So what about the effector part? So is IL-17 required? So we knew that global IL-17 receptor knockouts, we cannot vaccinate them. But we wanted to know, how are these cells working in the lung? And so we took advantage of some conditional knockouts that we made, and these mice are commercially available at the Jackson laboratory, but where we took IL-17RA flox mice and we crossed it to Tom Mariani's CCSP Cree line that he made at Rochester, and then we generated a Dermo-1 Cree line to knock it out in fibroblasts. And you can see when you knock out the IL-17 receptor in the club cells, in the conducting airway, there's a slight decrement in vaccine immunity, but it's completely lost when you knock out the IL-17 receptor in fibroblasts, suggesting that these cells are talking to lung fibroblasts. So we did RNA scope on these, and this is the cells that are elicited. This is prior to challenge, and you can see that they sit in the submucosa below the airway, and they're slightly reduced in the dermo-Cree mouse, and we think that this is we think that this may be due to less eyeball formation, because Troy Randall's group has shown that IL-17 receptor in fibroblasts is necessary for eyeball formation, but there's still plenty of cells in the submucosal space here, and this is again where these lymphatics are also channeling and potentially making IL-7, as the Flavelle paper showed. So that may be an important way for these cells to survive in this niche. So when we got this dermo-Cree result, we went back and looked at our single cell RNA-seq data in the lung, because it's even though we enriched for CD4 cells, there's a lot of non-CD4 cells in there, and we said, what cell expresses IL-17RC, which is the receptor for IL-17A, along with IL-17RA, and what cell expresses dermo-1 or twist2? And it turns out that it's a adventitial group of fibroblasts that are marked by collagen 1A2. So it turns out that Jackson Laboratory has an inducible Cree for that, and so we're in the process of seeing, are these cells talking to these adventitial fibroblasts that are around the airway for vaccine efficacy. So having trained at Pittsburgh and seeing a lot of immunocompromised patients, particularly with multi-drug resistant Klebsiella, one of the patient populations that are susceptible to Klebsiella are transplant patients. And we'd shown several years ago that Th17 cells are steroid resistant. So we wanted to know, are these cells resistant to FK506? And surprisingly, so the model here is we immunized mice, and then we gave them FK506 before the challenge period. And it didn't affect the total lung population, but it remarkably inhibited Th1 responses, interferon gamma. But the IL-17 responses were only about 50% affected. And this level of residual IL-17 was associated with significant vaccine immunity, even in the setting of FK506 immunosuppression, suggesting that perhaps these types of adjuvants can elicit protective responses even in the setting of pretty potent immunosuppression that we use to treat transplant. This FK506 resistance could also have implications for allograft rejection, because we see allograft rejection even in the presence of therapeutic FK levels, and maybe this FK506 resistance could also be harmful, particularly with a T cell that's recognizing an allograft or in autoimmune disease, potentially a self antigen. So we're actively trying to understand this FK506 resistance. It does look like there's 50% of these cells do not express the target for FK506, which is in the mouse, is FKB1A. But we don't know why that is, whether that's epigenetic or whether there's some other effect on that. So we don't have any data to understand what mechanistically why is this population FK506 resistant, but it is kind of intriguing that these cells are clearly different than Th1 cells, which are susceptible to FK506. So in conclusion, LTA1 in combination with a bacterial outer membrane protein can elicit these lung TRM cells, and these cells can elicit serotype independent immunity in the lung, and they appear to occupy a really unique niche in the parabronchial space, and they appear to communicate with lung fibroblasts for vaccine efficacy and not the conducting, at least not the club cells. Obviously, this would, there's obviously maybe signaling to non-secretory cells like ciliated cells or even goblet cells may be important, but at least I think we can safely say that the fibroblast signaling seems to be pretty important for their efficacy. And with that, I'd like to conclude. So a lot of this work was done in collaboration with the Norton lab here at Tulane, and then Naoki Iwanaga, who's actually a respirologist visiting from Japan, has done a lot of this work. And the single cell RNA-seq work was done with Keijing Zhang, and Alana Wanek was the bioinformaticist, and then Trevin is a master at maintaining all our mice. So I'll stop there.
Thank you, Dr. Kolls. I'd like to go ahead and have you guys put any of the questions that you may have in the chat box, and we'd be happy to raise those to the panelists here. I'll read them out as they come in. But I think to give you a little bit of time to formulate questions and enter them in, I can start off with one or two now. And I think that's probably, we can start off talking about COVID, because that's obviously a topic at the front of everyone's mind right now. And thinking about the various opportunities for adjuvants that would create some mucosal immunity for COVID, do you think that would be beneficial? Would that be a preferred mechanism of vaccination? And do you have any data that would directly look at viral vaccinations and adjuvants with any of the adjuvants you just addressed today?
Betsy, you want to take that one?
Sure. So if you recall, our studies with flu and LTA1 show reduced viral loads, quite, I would say, impressively decreased from just the flu zone antigen alone. So that would suggest that in some ways using an adjuvant can really help with decreasing that viremia. So a question always with vaccination is, are you reducing symptoms and clinical disease in somebody, or are you reducing just transmission in general? And I feel like adjuvants can really help direct immunity so that you get kind of a virus that will not be replicating in a host and will not be able to transmit to another person. So you're not getting that, even if you're not getting both clinical disease in the recipient of the vaccination or transmission to somebody else. Jay, would you like to add to that?
Yeah, well, Alexa, it's interesting. So last week in the New England Journal, there was a GWAS study on severe COVID, and they were all lung immune genes, so both dendritic cell genes, some NK cell genes, but one of the genes was CXCR6, which is a marker of lung TRM cells. And we don't know the direction, we just know that that CXCR6 was, a SNP in CXCR6 was associated with more severe disease, so it raises the issue, are lung TRM cells helpful or detrimental in COVID? And so I think we'll need to get more data on that. I know that like the CF Foundation's also trying to understand, are other diseases like cystic fibrosis at risk for COVID? So, but yeah, I agree with Betsy. I think one advantage of a mucosal route would be the fact that you can get both a pretty strong mucosal T cell and B cell response, and whether there would be some advantage of having that over traditional immunization subcutaneously, which is largely going to give you an IgG.
I would like to just also add to that, one things that adjuvants have been very attractive for is that in times of vaccine shortages, this dose-bearing effect where you can reach more people. So if you're trying to rapidly create a COVID vaccine, depending on the type of vaccine, and you have so much protein or inactivated virus, if you add an adjuvant to that, then you may be able to reach a lot more people with the same vaccine. So.
Yep.
Great. We do have a couple of audience questions here. First one is for Dr. Norton. And this person asked, Dr. Norton said that LTA1 was used intramuscularly, intranasally, and et cetera. How about subcutaneously? Is that possible?
Oh, yeah. So, there are smaller differences, maybe when you compare intradermal to intramuscular to subcutaneous, depending on the vaccine. But with these differences, they're very minor. They're not nearly the type of differences that you see in the injected routes than you see with mucosal delivery, such as comparing subcutaneous to intranasal or subcutaneous to oral. So yes, they would all be fine subcutaneous.
All right. We also have another question, doesn't specify if it's directed at Dr. Norton or Dr. Kolls, so I'll let you guys choose how you would like to address it. The question is, will the adjuvant increase both IgG and IgA levels in specimens, or is it one or the other or both?
So this is sorry, Jay, I'll take this. What you see with adjuvants is that there's always a combination effect with the antigen that you are co-delivering it with. So in general, these adjuvants give you an IgG and an IgA response. The magnitude of that response can change depending on which antigen you deliver it to and what route you're using. So if your target is really a high level of IgA, then typically the best response that we see would be intranasal or oral, but you would still see that with injected routes. However, the antigen starts to be a lot more important for the injected routes for the magnitude of IgA. And I'll just give you an example that with tetanus toxoid injected intradermally with DMLT, we see a modest amount of fecal IgA after vaccination. This is also true of polio injected intramuscularly. We see a modest amount of fecal IgA. If we use, in contrast, an ETEC fimbriae tip adhesion called CFA-EB with DMLT, there's a huge amount of fecal IgA that we detect. So the antigen really can be critical in combination with the adjuvant.
Yeah, and then I'll say in the setting of the Klebsiella work, we see both an IgG and an IgA response. The IgG actually stains the surface of the challenge strain as well as the parental K2 strain. So even despite the big mucoid capsule that's on hypervirulent K1 strains, it appears that OMPX is a accessible antigen. And then the IgA response is actually really greatly diminished in our fibroblast receptor-specific knockout, but not in the club cell. So even though we've shown previously that club cells express the IgA transport receptor, but presumably IgA is intact in those mice because it's being transported by other cells other than club cells. But it's really diminished in the dermo-Cree line. So and we don't know whether that's a transport issue or whether there's also reduced numbers of IgA-produced antibody-secreting cells in those mice.
Do you find that this is something that you can predict going into the experiment, or is this something you have to empirically determine, such as where you're going to see the best IgA response for which protein, or is it predictable?
I would say it's fairly predictable that if you do the mucosal immunizations with these adjuvants, you get a good IgA response. It becomes a lot more empirical when you start talking, in my experience, with the injected routes. It becomes something that you have to test.
Yeah, and I think, Alexa, that's a really important point too. Is like if we're really going to move the field forward with inhaled vaccines that potentially work through local antibody or local T cells that particularly that don't circulate, right? I mean, lung TRM cells don't really circulate in blood. We need to have non-invasive measures of how do we know that we actually elicited that response in a human? So potentially antigen-specific IgA in sputum could be a really effective biomarker to try to detect these immune responses in clinical trials.
That does bring up another question I was thinking about as far as intratracheal or inhaled delivery of vaccines. That is something that, we have a FluMist vaccine, a live intranasal vaccine that, several years ago, there was some concern that it was leading to, particularly in some decreased efficacy, even though there might have been better uptake because it didn't involve a shot among children. So that one was kind of de-emphasized, but what kind of work have you done to look at intratracheal administration or inhaled administration of some of these vaccines with adjuvants?
So we've compared sub-Q, sublingual, and in what we call intratracheal. So the data I showed was all with intratracheal. The sublingual approach and the subcutaneous approach, at least with this antigen adjuvant combo, was not as effective. We were planning on initiating some non-human primate studies where we could give a better intranasal model that would hopefully be closer to humans, but of course COVID hit, so that got put on the back burner. But so I think we need to compare intranasal versus inhaled routes in animals models that more closely mimic the human airway.
But I would say, Alexa, to your point about the FluMist was a great one because it was de-emphasized for a few years, and then they found that the use of it, especially as school-age children, was lower, and so they brought it back in this most recent flu year to have better, vaccination kind of adoption in different communities. And so I think mucosal delivery, needle-free delivery is a great option for children and for many different populations. And so making sure it's safe is, of course, a paramount importance, but there's options to do. If you don't want to do directly into the nose, maybe you can do an inhaler to get to the lungs. Maybe that's better for reaching the TRM cells that Jay's talking about. I mean, I think there's a lot of compelling reasons to investigate this further.
Certainly, many of those, the best vaccine is the one that gets given, and so you don't want to decrease the amount of. You want to perfect your vaccine and then decrease the uptake because you've made it very unpalatable for your patients, and that's particularly true in pediatrics. But there's also a lot of adults who as well will avoid vaccination because they don't like needles. The other, basically, other concerns would be related to, how can we, looking at the timing of vaccination and what vaccines can be given with other vaccines either at the same time or, within a certain time span of each other. We have, with live vaccines such as MMR, we have to be very careful to not vaccinate within about a month afterwards with any other inactivated vaccines. It seems that you get decreased immunogenicity of the second vaccine if it's given too recently to one of those live vaccines. Do you have any other similar concerns with some of the adjuvants that you've mentioned here? Are there any limitations on how they can be formulated, when they can be given, et cetera?
I think they're good candidates to be used with other vaccines, particularly DMLT in clinical trials with a combinatorial heat-killed ETEC vaccine was shown to, if the responses were really high to specific antigens, it didn't enhance those responses, but to antigens that had low responses in adults, DMLT really help to boost those responses. And to me, that's promising because it means if you're combining the vaccine either close to a schedule with another vaccine or even giving this DMLT adjuvanted vaccine at the same time, that it's probably not going to diminish the response to other antigens, but just help the response to the antigens that are at low magnitude and need to be boosted up. Jay, would you like to comment?
No, I mean, I agree. I mean, I think, obviously it's probably less of an issue in adults where, which the bulk of the vaccines are given in the first, 5 years of life. So it's more relevant to childhood immunization. And, that'll have to be carefully considered in that population.
In adult medicine, we see it a lot in people who are traveling and need, boosters for MMR or need yellow fever vaccine when that becomes available again to us more widely in the US. So, yeah. Do you foresee any limitations on which organisms you might be able to employ some of these adjuvants with, or how do you think about the pros and the cons of each of these selections and where you might go with other pathogens?
Well, one concern we had, and we've actually gotten comments on grants we've submitted, is effects on the microbiome. And so I failed to mention we did look at fecal IgA in our Klebsiella experiments, and with the route of administration, we don't see any fecal IgA. So it does appear to be specific to the lung. I mean, I think we're prioritizing encapsulated bacteria. Klebsiella, Streptococcus pneumoniae, those pathogens. I think one of the concerns I have is obviously the duration. What's the duration of vaccine efficacy, and how often will we have to boost? I mean, those are things that those are long-term experiments, and they take a while to figure that out. But those are important issues to try to translate this into the clinic.
Okay.
I'll mention one other thing too. Well, now that we have drugs for cystic fibrosis, maybe this is less of a concern. But we think that these cells they need relatively normal lung architecture to work, right? So, in CF, there's plenty of Th17 cells, yet they don't clear the pathogen. And we showed a few years ago, one of the things that IL-17 does is regulate bicarbonate transport in the airway, and that's absent in CF. So one possibility in CF is that you generate these immune responses, but they're just frustrated because you don't have normal mucosal immunity. Well, in a lot of chronic lung diseases like non-CF bronchiectasis, COPD, maybe uncontrolled chronic asthma, these types of vaccines could actually make disease worse because of the architectural abnormalities that occur or the epithelial abnormalities that occur. So I think having going into a healthy population that has relatively healthy lungs without these underlying disorders would be the first population I would go into, because I would be concerned about having unanticipated side effects in patients with chronic lung disease.
Yeah, and I would just say, I mean, it's a vaccination, so there's always a risk to benefit with any vaccine. I mean, because you are doing something to the host. So being really careful in the way that you target your population and maybe select out people who would be at higher risk for unwanted side effects, and also just working, I think, with regulatory agencies kind of early on in your design process to make sure that you are appropriately dealing with that and thinking about it if you're starting with these adjuvants in preclinical studies. What's nice is that DMLT has been used in enough clinical studies right now is that there is safety information with oral injected routes, and as well as there's some information about transcutaneous, sublingual. There isn't as much information about intranasal with LTA1. And so that moving forward, I think would be an obstacle to overcome, just because anytime it's the first time, you have to make sure to overcome it. Given that there was some past history with the AB5 molecules having some unwanted side effects. So it's definitely always important to keep in mind, and then, as I said, work with regulatory agencies when you get to that, working on that process.
Yeah, certainly with an inhaled or an intratracheal vaccine, you'd want to be very careful about any sort of generating a too robust and inflammatory response, and almost in those, especially those susceptible to asthma or COPD, who might have a flare or an exacerbation of their disease, just as a response to the vaccination. Certainly something to be mindful of, and I think you've obviously are doing that as well.
Yeah.
I think we don't have any additional questions from the audience at this point. Did either of you have anything else that you wanted to make sure we address today with the group online?
I think we covered everything. I'd just like to thank the ATS for pressing on with the program despite the challenges of 2020 here.
Mm-hmm.
Yeah, I would echo those sentiments.
Yes, they've been remarkably agile in switching around to a virtual meeting with just a couple months notice, so that's wonderful. So I think with that, I'll probably remind everyone that we would appreciate your feedback, and I think the QR code is visible to you now. If you want to use that to fill out an evaluation for us, we'd very much appreciate it. And I'd just like to thank again the ATS and those of you who joined us this afternoon.
Great. Thank you, Alexa.
Yeah. Thank you.
Bye.